Okay, so I want to begin our new unit on biology by introducing something that actually has nothing to do with biology, but hopefully you'll actually see the overlap in a few minutes here. This is a thought experiment. Have you guys ever heard of the phrase a thought experiment in the sciences before? You're nodding. Are you nodding? Would you mind sharing anything you know about the term thought experiment? Schrodinger's cat is a great example of a thought experiment. Has anybody ever heard of Schrodinger's cat? Okay, so for those of you who have or haven't, I'll give you the basics of explain some quantum physics, which is super awesome stuff, by the way. Take physics. Okay, anyways. Schrodinger basically suggested this scenario. Imagine putting a cat in a box, putting some radioactive material in it that may or may not decay. The radioactive material would, in theory, then kill the cat or maybe not kill the cat. And then you seal it all up inside of a box in which you can't see anything in. And then he basically poses the question, is the cat dead or alive? And your answer to that is, well, I don't know. Has the radioactive material decayed or not? And you say, we don't know. And so Schrodinger's point basically was that the cat is both simultaneously dead and alive until you open the box, at which point one or the other happens. I actually even have some special Schrodinger's... Yeah, there's Schrodinger's cat mugs that one of my students bought me way back when. Okay, now, I don't need you to understand the Schrodinger's cat point. I can talk with you more about that later. But it's a thought experiment because it's just something interesting to think about, but no one's actually going to do this because putting a cat in a box with radioactive material is horrible. But it's an interesting discussion topic. Does that make sense? But in no way should any scientist ever perform that experiment. Plato's allegory of the cave is similar in that way, in which it's a horrible scenario to consider, and hopefully no one ever does this ever and has to live through this. So please don't go home and tell your parents that Chris suggested that we do something like what I'm going to tell you about here, because that's not the point. Plato's thought experiment was basically as a way to try to illustrate and describe some sort of more profound idea. Does that make sense so far? So it's a thought experiment. Here's his suggestion. He says, hypothetically, imagine that there is a civilization out there in which slaves have been captured. Okay, you can see where I'm going with starting on this. I'm not advocating for slavery whatsoever. Okay, but imagine that there's a society, a civilization in which slaves were captured, and they actually were forced to live underground. They are prisoners. Can you guys see where it says prisoners down there? They're prisoners, and this captive race of which there are prisoners and there are people who take care of the prisoners, I guess. They're captors. The prisoners have lived underground so long that they've never been outside. They don't know what sunlight is. called it a nucleus, but it should have been called an atom. We just assigned a random name to a lot of stuff in biology. We said, well, I see this. Call it that. Call it that. And that's a lot of where biology begins. Now, that's not all of biology, but a large part of biology is basically learning about stuff that we can't really see, but like we know it's there, and we're assigning a name to it. And your goal in biology, for the most part, is learn about this new topic, assign a new name to it, and so that's a large part of biology. Well, a large part of grade 10 biology is learning about all the different parts of the body. Right? So you'll learn about the fact that we have these things inside of us that I can't see. It's really unethical to cut a living person open. Take a look inside their body. Now, we do know after they die. We do MRIs, we do CAT scans, we try to see inside of their body. Is there really any purpose to why we called it a lung? We have called it something else, and it wouldn't have really made any difference. Yeah, we could call them breathing boxes, and that really, like, you could call it a blood pump. That wasn't a heart, right? So, anyways, I like Plato's allegory of the cave to begin with because I think it fits well. Now, by the way, Plato's allegory of the cave was I guess, come up with this. You know the name Plato? Plato was a Greek philosopher. So, like, we're talking about a guy that's like 4,000 years ago. So he did read his allegory in regards to biology. Like, the fascinating how weird is that? That you're now seeing dinosaurs rather than shadows. I like using it for biology, like I said. I like to make now, if you guys ever seen the Magic School Bus? We're actually going to walk down the Magic School Bus. Have you seen it? The one where they go and live at BB3. We don't actually have the ability to shrink down on the Magic School Bus and go down to the size of little bitty and go inside of a cell. That being said, we do have some evidence of cells. They don't like to go to cells. And we can just give them names. Like biology is all I took biology last year. Many of you are probably going to go take biology and biotechnology. But I like this fact of just biology and biology. Not the reason why is the order in which I teach things. We started with chemistry. Chemistry is the study of matter. Chemistry is the study of matter. Physics is the study of take that matter and give it some energy. Biology is the study of take that matter with some energy and make it become alive. The study of living things. And I think I actually have that on my next slide. So do we go to the slide right now? That's really the study of biology. We now take our matter. It needs some energy, and it's now considered alive. And that's one more step. It's sitting on the desk. It is matter. It's chemistry, but it's sitting there. It's not really physics. It's kind of boring. I throw it. Or something else. I get it moving. I make it use some energy. I turn it on. Now it's using the battery. But is my calculator alive? Right? And so that's now the study of biology is now we take those same two ingredients, we add the third one. And so here are the 5 requirements that science as to what makes something alive. You guys can read. I don't do this a lot, but I'll read off my slides here. Number 1, they're comprised of cells. Okay, we're going to talk about that over the next couple of classes. Cells are basically the smallest building blocks in the biology universe that are what we make things consider alive. It uses energy, so it still has that physics overlap, is that there needs to be some sort of energy to make them move. Cells need to do one of these 2 things. Or next block. Cells need to grow, develop, or change over time. Okay, cells don't just stay in one spot. spot. If it doesn't grow and get bigger, if the cells don't multiply, it's not really alive. It has to reproduce, which is very similar to number 3. Sometimes grow, develop, and change means make more cells, procreate, have a kid. So, if you don't have the ability to reproduce, then eventually that species will die out. And then finally, it has to be able to adapt and respond to its environment. Okay? So, let me go back to the slide I skipped here. Back to biology, it's all about things that are alive. There's botany, ecology, genetics, zoology. Do you recognize any of those words? Do you know what they mean? Anatomy, basically the study of the human body. So like I said, talking about what lungs are, what does a botanist study? Yep. Ecology basically just studies like environmental factors, how that works. Genetics, basically all about genes, the structure of like what is that even smaller building block? What are chromosomes? How does sperm and egg come together to make a new creature? Right? Zoology, animals. Okay. This is the word that I was really going for with Plato's cave. What biologists really study is structure, function, growth, origin, evolution, and distribution. But we're going to spend a lot of time on this word right here, taxonomy. And what taxonomy basically refers to is it's the science of defining groups based on similar characteristics. It's basically studying the things. You ever heard the two-word phrase Homo sapiens? Right, as a way of describing like humans as one of their taxonomic classes. A wolf is a Canis lupus. The canis coming from like canine for the dog family. So a lot of biology put names on things. I'm going to show you a quick little YouTube clip here. Talking about some history about how cells have been discovered. So, start with this.
Guy who really loved loved all aspects of science, so he dabbled in a little bit of everything, including physics, chemistry, and biology. Thus it is Hooke who we can thank for the term the cell, as he was looking at a piece of cork under his microscope, and the little chambers he saw reminded him of cells, or the rooms monks slept in their monasteries. Think college dorm rooms, but without the TVs, computers, and really annoying roommates. Hooke was something of an underappreciated scientist of his day. Something he brought upon himself as he made the mistake of locking horns with one of the most famous scientists ever, Sir Isaac Newton. Remember when I said Hooke dabbled in many different fields? What? A breaking book on how planets move due to gravity. Hooke made the claim that Newton had been inspired by Hooke's work in physics. Newton, to say the least, did not like that, which sparked a tense relationship between the two that lasted even after Hooke died, as quite a bit of Hooke's research, as well as his only portrait, was misplaced due to Newton. Much of it was rediscovered. Last part of the cell theory, that cells come from pre-existing cells. Leiden didn't exactly subscribe to that thought, as he swore cells came from free cells. That's when another research.
Or like, probably just stop there. Covered one of the first ones. Would want to know. Video. Only appears. We kind of intuitively picked up and learned that in order for there to be something living, it had to come from essentially parents or other cells. But creature. Imagine that you left some like rotting meat. I mean, you cook your steak, you eat your meat, there's some leftovers, and you leave it sitting there. And you leave it sitting there. And you leave it sitting there. We now recognize that it's going to rot. It's going to grow bacteria and start to grow moldy. But what scientists at that time were seeing was, hey, there's some flies starting to show up nearby. The meat is turning into flies. The wheat is turning into frogs. That was their theory. I mean, it kind of made sense to them. To us, it sounds crazy because we know better. But at the time, they thought that like living creatures were just randomly, spontaneously growing out of non-living material. We now recognize that the food was spoiling and it was attracting creatures, not producing. Can I say that one more time here? We now recognize that the spoiling of food was attracting creatures nearby, and it wasn't making them. Frogs weren't just randomly showing up in like a barn. They had found a new habitat to live in. And so I think one of the stories is about corn husks. A farmer who has a field full of corn, the corn's all been picked, it's all sitting in a barn, and all of a sudden they discover that there's frogs in it. The frogs just came for the corn. They didn't clue into that. Does that make sense, though? Okay. So that was the theory, spontaneous generation. Okay. So the first person I want you to know about, his name was Francesco Redi, and he's the type of wrapping, right? Or it's a little bit breathable. What makes something a controlled experiment? You need to have a controlled, a manipulating, a responding variable. You guys familiar with that vocabulary, controlled, manipulating, responding? We've done this before a little bit. Control is what you keep saying. So in every single one of these experiments right here, we use the same type of meat. It wouldn't be fair to use pork on this one and then beef on this one, chicken on this one. So use the same amount of meat, use the same type. Right, yeah. I mean, technically did 3 experiments on this, but does that make sense though? Pair A, they're like molecular agency. This one's open to air. Okay. What was the responding variable? Whether or not bugs grew, right? Yeah. If bugs grew, that's the responding variable. So I know that sounds kind of silly to us because you guys probably know this vocabulary is responding variable, but in the year 1868, this is sometimes thought of as the start of the scientific method as to how do we actually realistically do an experiment. Keep in mind, chemistry really hadn't been developed at this point yet. Most of the stuff we talked about in chemistry didn't happen until well after this point. Most stuff in physics actually didn't happen until after this point. So this was a very, very conservative time. Follow that up. Another scientist, you might actually recognize his name. His name is Louis Pasteur. We actually named pasteurization of milk after him. He hypothesized there were microorganisms in the air. said, oh, okay, you covered it. And covering it seems to solve the problem. I mean, how many of you know this now? If you have food, put a lid on it. And furthermore, probably put it in the fridge. Right? But like, if I have a sandwich and I leave it sitting out on the counter, it's going to cause the bread to kind of become crusty after a while. If I have an apple and I take like 3 bites out of it and I leave it sitting on the counter, it's going to start to brown. Right? Like, we get this. They did this time. They hypothesized there were microorganisms in the air responsible for the growth. So, he made his own experiment. This was actually, this picture right here was really another picture of Francesco Redi's experiment. So let me show you Pasteur's experiment. He did this. He basically made soup. He made a nutrient broth, something that would have been nutrient broth right here. He boiled it. What do we now know? What does boiling do to something that might have had microorganisms? What would it do to it? Like, let's say that we have a boil water advisory. We haven't had a lot of those probably. But you guys know that, like, if you need some water and it's not safe to drink, one of the possibilities is you boil it. Why? Well, because it would kill any. Well, we now know the term is bacteria and germs and viruses and stuff like that. It would in theory kill all of the stuff. So if you boil this broth right here, it should, in theory, remain free of microorganisms. But what's air is able to get inside, right? This flask right here, will you from air getting inside? No, because I mean, air is totally able to get inside. He discovered that if you have this S-net flask on it, nothing happens. Now, take this little S-net off, and all of a sudden it has a regular beaker, like microorganisms through inside. What he actually discovered is that a whole bunch of growth of microorganisms actually took place right here at the bottom of the S of the flask. They didn't show up in the broth. They have this little S-net flask right here. What ended up happening is the bacteria and microorganisms that we now know about, they weren't in there. They were, right? When I go, shoot! You guys have all been taught by your parents to cover. Of like bacteria. Is that a fair word for me to use right now? Can we just call it mold? It wasn't air that caused the problem. It was the bacteria in it. That's why when you saw the bacteria, the mold in the air, you realized it's not air that's the problem. So now we've also got to go back and check what scientists are doing at the time in terms of trying to see these microorganisms. You and I have really limited. We can't see everything. So the first people that we kind of give credit to playing around with microscopes and magnifying glasses are the brothers Andrew Zacharias Jensen. And the first recorded known microscope that he tried in 1995. One of the challenges in looking back 400 years is that I don't know if you know this because there might have been some other random dude who was playing around with glass and discovered a microscope, but like we don't have a record of it. So we kind of give credit to the Canadians. They made a microscope for the first time. And as the YouTube video kind of showed, there was actually a real boom at the time. So, telescopes, magnifying glasses, microscopes, everyone just wanted to look at stuff under a microscope. One day, I brought a microscope home from school to show my 7-year-old. And I don't know if you guys have ever got a chance to play with microscopes, but like it's kind of fun, isn't it? Right? You're like, ooh, what else can I look at? Let's look at my hair. Let's look at this bug. Right? Well, Robert Hooke was studying cork. Do you guys know what cork is? Yeah, it's like what you cap a wine bottle with. The reason why is that it swells up when it gets dry, right? So that it actually seals up the wine bottle. So they drank a lot of wine back then. So he was. This sounds really bad. Maybe I shouldn't say this one on my video, but I'm going to. So he's drinking one night his bottle of wine and goes, I wonder what the cork is made of. Slices a little piece of it off the microscope, and he sees square boxes. And I want to go back to Plato's allegory of the cave. He has no idea what square boxes are. He says, you know what they look like? They look like where the monks and priests live in a church, where they get like their own little room. And that room is called a cell. So he's like, I call it a cell. Remember how I talked about Plato's allegory of the cave and how we just assigned a random name to it? Isn't that crazy that we call it a cell? Because one random guy, and I don't know that he was actually drinking at the time, right? But had some wine, had a cork nearby, took a look at it, and said, hey, it looks like this. Let's go with that. Oh, that's that stick. Isn't that like weird? You know, because we could have called them like living boxes. We could have called them anything we wanted. Here's what Robert Hooke saw. Okay, I don't actually have evidence he was drinking. That's why he said, hey, it kind of looks like all those rooms that okay, around the same time, I think I saw this in the YouTube video, a guy named definitely playing around with microscopes, and it's just gross. He's like scraping stuff off the microscope, and what's different about that is that he actually saw living cells. Because cork, being made out of wood, is dead. It's no longer alive. Right? It swells up, but it's not actually alive anymore. Well, Van Leeuwenhoek basically had taken bacteria and scum off of his unbrushed teeth. Allegory of the cave. Call it a word. Another person I'd like you to know about, Robert Brown. He was the first person to identify that there were actually smaller structures. A lot more stuff inside. You won't be able to see mitochondria, cytoplasm, or lysosomes, or any other things you might have heard about. Cell features. Wait, so fast forward a couple of years, and scientists are still trying to figure out the biogenesis thing. And two scientists who saw on the YouTube videos decided to collaborate with Schleiden, I think, actually. And one was working on plants, one was working on animals. And they both had the same urethra multiple. Which to us sounds really dumb if you're like, obvious. But at the time, they didn't figure it out. One guy was like, I have been studying animal after animal after animal, and guess what? They're all made of cells. And the other guy goes, I have been studying plant after plant after plant, and guess what? They're all made of cells. Living things are made of cells. That's basically their thought process. At the time, they didn't realize that living things are made out of cells. They finally put together that organisms are made out of cells. So this is really one of the main points of what the cell theory, which I'll tell you on the slide here, is that in order for something to be living, it has to have cells. But those cells, they have to come from other cells. He's kind of the one who kind of tied it all together. Heard about these two scientists corresponding. I don't know how the story goes. I kind of imagine cells. And so here's a basic timeline of the different scientists and what they accomplished. Rudolf Virchow is the one who stated, this is the Latin, I don't know that I'm saying the Latin right, it's omnis celia is celia. It basically means cells create cells. That was basically his thought. Cells make more cells. So here's our summary then. What do we now know as the result of a few hundred years of some scientists randomly naming stuff that they were looking at under a microscope? 3 things. Number 1, all who are alive. The cell is the smallest function of life. There is nothing smaller than a cell that we can consider alive. Is there stuff inside of cells? Yeah. There's a nucleus, there's mitochondria, you guys have heard of some of these things in your previous courses, right? Okay, there's cytoplasm, ribosome, cell wall, cell membrane, all that stuff. None of those things are on their own enough to be alive. But a cell is alive if it's made of fluid. And the final one, like Rudolf Virchow said, a lesson on your phone as well. There's 5 multiple choice questions. 5 things you hope to learn. Allegory of the cave. No, I'm not advocating that we trap human beings underground, raise generations of people who haven't seen daylight. But like the whole point of Plato's allegory of the cave is that we can't even see. And when we can see it, we see it through a microscope lens. We're not even seeing the real thing. We're just seeing like the effects of it. Kind of like in Plato's cave idea there. 2, spontaneous generation, the idea that things just randomly showed up. Do you understand the 2 experiments, Francesco Redi and Louis Pasteur, and how their controlled experiments helped to really prove that like bugs didn't just randomly show up? Okay. 3, do you understand controlled manipulating responding variables and how in 1665, whatever the day was, that they didn't do experiments a lot before that. This is controlled experiments we now take for granted, like do everything the same, change one thing, right? That really helped them to prove that. Here's a whole bunch of names we learned about: Janssens, Hooke, Van Leeuwenhoek, Brown, Schwann, Schleiden, and Virchow. What did they do? They all kind of contributed a little piece along the way. And then I'm going to leave this up on the slide there. If you'd like to, feel free just to copy it out. But the 3 main points of the cell theory: cells, if you're alive, you're made of cells. Nothing smaller than a cell that's alive. There is stuff that's smaller than a cell, but not that's alive. Okay. So, yep. Cool. Okay. Work on those reflections till the bug goes. Thanks, everybody.